I'm going to talk today about, I'm giving a tutorial talk actually, but I will end up talking about my work because, of course the tutorial is still stuff that I'm very interested in. So today I'll be talking about harnessing data geometry for biomedical insight. So I guess overall my talk will probably be quite different from a lot of the other talks. I identify myself more as a data/computer scientist / applied mathematician who's very motivated by problems in biomedical data science. So basically what kind of motivates some of the methods that I like to think about and the techniques from data geometry that I'd like to tell you about today is basically the fact that we have a very highly complex entity whenever we look at any biomedical system. It can even be a single cell, but this is a map of gene interactions in a particular cell type, and you already see that it's very canonically complex in the sense that it has a lot of interacting components, and they're interacting in pretty strange and nonlinear ways. And so, as a data scientist, I'd like to know if we can discover insights about this from the data. Okay, and so right now there are gobs and gobs of data being generated. Bogdan, for example, was telling you about gobs of data on potentially patients or other populations. We also have a lot of data in genomics, now in the form of single cell data, which is very big in that it has a lot of dimensions as well as a lot of features, like thousands and thousands of features, and each of which are counts of genes. So at the end of the day, you can think of these as a huge matrix, and even a tensor if you have many, many samples or cohorts of this kind of data. And some of the fundamental challenges that come with biomedical data, sort of as part of the territory, is a lot of it comes with a lot of noise. So this is both due to measurement artifact, but also just measurement sensitivity. And the bigness of the data in terms of dimensions and in terms of the number of observations can make it difficult to really understand what's going on overall. And so these are fundamental challenges in terms of just exploring the data without necessarily even knowing anything prior. So a lot of the techniques I'll talk about are sort of first from the get-go kind of techniques. So if you just arrive and you get a huge matrix, then what do you start doing with it? And how do you get any insight on this data? Another thing that compounds the difficulty of this is these days you can get completely different sorts of measurements all taken on the same entity. But because they're part of the same entity, they must have something in common, and you want to extract that out. And maybe another thing that we maybe sometimes forget is whenever we measure any kind of data, it's usually a static snapshot data point of something that's continuously evolving and changing. And the same is true of DNA sequencing. So if you measure somebody at a particular point, you might not see the mutations they're going to get tomorrow, etc. So some of the challenges are reimputing the dynamics, also from the static snapshot data. But something that helps save us from some of the difficulty is actually a hypothesis that scientific data, and in particular biomedical data, is often sampled from an underlying manifold, or at the very least, this is a very useful model for this kind of data. But of course, often so these data points are from some underlying manifold that you don't necessarily know what this manifold is. And moreover, they're sampled with noise, so they're somehow splayed apart from the manifold. So you might be asking, okay, what exactly is a manifold? So you can think of a manifold, intuitively speaking, as sort of like a sheet, like a 2D sheet that's coiled up in 3D space. Or if you really want to think about single cell data, maybe a 20D sheet coiled up in 20,000 dimensional space. And so the interesting thing about it is that locally, manifolds resemble Euclidean spaces. So locally, you could use Euclidean distances, for example, to find neighboring points. And then you can create sort of longer paths that create sort of a mesh over this data. So you might be asking, okay, this is a cute hypothesis, but why is this true of scientific data? Well, the first thing I'll say is, by this point in time, my 6th, 7th year of faculty position working with these techniques, I can say the proof's a little bit in the pudding. Whenever we've used manifold models, they've actually produced pretty good results. and not just me, several of my colleagues, mentors, etc. But I would say there's sort of a philosophical or logical reason you can think about it this way too. So the picture on the left of all the features you measure in your data, whether it's genes from your cell, are all sort of uncorrelated or uninformative of one another. But this really can't be the case because they're all parts of the same entity. For example, if you know the location of my right hand, you have a very good idea of the location of my left hand, right? It can't be in the back of the room because I have to function as a person who has two hands and things like that. So what this does, the fact that these entities are all correlating and talking with one another, reduces the dimensionality of the space. It restricts it to a subspace. And the reason why there's smoothness and why you could actually learn it is A, we have a lot of data points that can fill this space, and B, these entities change gradually. So cells, you and I changed very gradually from one cell to the trillion cells we are today. Did not happen like in an instant. So whenever you measure these systems, you often get a pretty good sampling of the whole space. So that's kind of why it might be true philosophically speaking. This is a tutorial talk, so let's go to what I really mean by data geometry. And geometry, you might think of stuff you learned in grade school, and that is all part of geometry. So geometry, actually I looked this up today, comes from the Greek word geometria, where it says land measure. So it's basically a branch of math that's concerned with the properties of space, like distance, shape, size, curvature, etc. And so the shape of data can actually tell you quite a bit about something. The thing I have on the bottom are actually fate plots of real data. So it's not synthetic data, but the shapes are very different. They tell you something very, very interesting, which I'll come to later on. So the first thing you might want to do if you get such a huge matrix is actually try to see what the shape of this data is. Okay, so one of the important properties of our data set is that you can actually model it as a metric space. A metric space is a set like your points together with with a function, and the function has to satisfy the usual distance properties, so identity, symmetry, and triangle inequality. Okay, so once you've found a metric, you've kind of solved half the problem. So what this means is you can start thinking about your data points in relation to one another rather than in terms of their large feature space. And so this is one of the ways where thinking about data geometrically really helps us. And really, the idea of thinking about distances between data points forms the basis of most unsupervised machine learning methods, including kernel methods, clustering, which you saw several times, visualizations, embeddings, autoencoders, etc. Autoencoders, for example, you may not have to explicitly put in the distance, but nevertheless, their latent space learns one, and that can be useful. So I'm just going to give you examples of a few kinds of metrics that we'll actually use in order to make sense of biomedical data. One is just a Euclidean metric. This is the one that you're used to. It's just a Euclidean distance. And so it's the square root of your x values squared, y values, taking the difference of the x values, y values, and then squaring and square rooting them. Another one that we'll talk about is a metric that's not on single data points, but it's actually on distributions of data points. And this is when you get a tensor instead of a matrix, you have a whole bunch of data points on one person and then a whole bunch on the other, as if, for example, if you took single cell data on many different people. And here, this is called the Wasserstein or earth movers distance metric, which tells you the transport cost of moving one distribution to match another distribution. And what you see there is actually a transport plan. So the bins here are divided up and transported in different ways. And the Wasserstein metric is measuring the optimal of those kinds of transport plans. Feel free to stop me if you have any questions on any of this. Another metric that I'll talk about is a geodesic metric. A geodesic metric would be a metric of how far something is on the manifold. So you see this ladybug here, and it's actually just kind of walking on this manifold. And this kind of distance becomes very useful when you actually model your data as a collection of manifolds, is how can you get from one data point to another. And this turns out to be a really useful distance to preserve in biomedical data. So let me tell you about one other concept, which I started talking about again. So I talked about a data manifold, but there's a concept of a Riemannian manifold. That's a smooth manifold where at each point there's a tangent space defined, and this gives rise to a family of inner products called the metric tensor. And these can be used to integrate it out to give geodesics on the manifold. So we want this kind of idea that you can have local distances that you can integrate out or walk along to get distances between distal points in your data. And we're going to use this also. And finally, one other sort of very geometric concept that you might not have thought applies to your data is actually curvature. So your data has curvature. If it didn't have curvature, it would be flat in the ambient space in so many dimensions. And curvature can actually be used as a feature. And this is one of the functions of data geometry, but also topology, to come up with these kinds of features that can describe your data. And then you can feed these features upstream to a classification or other kind of system that can reason about them. So the concept of curvature, mathematically what I'm showing you is Gaussian curvature. You can think of what circle osculates or kisses your curve at each point. And if it's a very small circle, that means you have high curvature. So it's inversely proportional to the radius of the curvature. And here, this is a flatter region, so you can fit a bigger circle here. And so this is one of the concepts of curvature, and you see even in this 1D data, the curvature constantly changes. And actually, curvature can give us a lot of information about biomedical systems too, because it says something is changing or bending in the shape of the data in an interesting way. So this is that was my short intro to concepts from geometry. So distances, geodesics, curvature. And so if you want me to go back to any of these concepts, let me know. But this is the outline of sort of the rest of my talk. I'm going to talk about how to sort of learn the dimensions of the manifold from the data, and then how you can use data-driven metrics to perform analysis on the data, including visualization and multi-scale analysis. Then I'm going to switch from talking about the shape of a single data set to multiple data sets. And in there, I'll talk about how you can find transport paths and differences between multiple data points, multiple data sets. Okay, so the overview of the method is as follows. So a sort of basic canonical way to learn the manifold of data is if you have data in 2 dimensions, you can compute distances between the data points. But as I said, these Euclidean distances are not that reliable if you assume you have data on a manifold. Only locally Euclidean distances are reliable. So you localize this using an affinity kernel. Now you have something like a soft nearest neighbors graph. Here it's even thresholded. This starts to follow the shape of the data already, so you can see something about the shape of the data. One thing that's very useful for finding geodesic distances on this is to see, typically if you had no noise, you could just find a shortest path using this kind of a graph that would tell you what is the distance between those. But it turns out this data has a lot of noise. So instead, what we define is a transition matrix that says, what's your probability of jumping from one point to another? And that's inversely proportional to distance, and that can give you a probability of walking from any data point to another, which can help you derive a geodesic distance on this learned manifold. And downstream of that, learning these, there's a lot of things you can do, since basically distances between points form the basis of clustering, visualization, etc. Okay, so just to go a little bit deeper into the steps that I mentioned. So I start out with a matrix, as I said, and I'm thinking of single cell data here, so I'm saying cells by genes. Then you can use Euclidean distances here to get pairwise distances, or you could use other distances if you felt those were better suited. Then you would compute affinities. But you see, the affinities, like the distances, still have these paths that are connecting between data points that aren't on the density of the data, so they're not following the shape of the manifold. So the thing that really helps us with this is this diffusion operator, where we've normalized the affinities by their row sum. And that gives you a Markov matrix, and you can raise it to take t-steps walks on your data. And that gives you something very clean where most of the paths are on your data. The diffusion operator actually can be computed de novo on any graph if you wanted to perform a random walk on it. So if you even had a 0-1 graph, you'd row normalize it and you'd get walk probabilities that describe how if you started at one vertex, how you would transition to other vertices. Okay, and the really interesting thing about this operator is it's been proven actually many times that this operator is a discretization of a heat operator. So it discretizes this idea that you have one drop of heat and it's spreading through the manifold. So it's just a discrete version of running this kind of heat diffusion or heat flow on your manifold. And so one of the classic techniques for manifold learning, indeed, is diffusion maps. They were invented actually by my colleague at Yale, Rafi Koifman, and his student at the time, Stefan Lafon. That one is pretty easy for me to describe now that I've described the diffusion operator to you. The diffusion operator is this matrix of transition probabilities from one data point to another data point. The diffusion map is an eigendecomposition of this, and the new diffusion map representation of every data point is the value of each eigenvector weighted by the eigenvalue. And so the diffusion map representation is a new space for your data. And in this new space, the Euclidean distance in this space is equal to a geodesic distance. It's equal to basically the distance between 2 points tells you how random walks arising from those points diverge or deviate from one another. And so this enables a couple of different things. One is, I'll go in the opposite order of the slides I had. The first one is spectral clustering. So if you have these new diffusion, if this new diffusion embedding, it somehow unrolls your data so that you're getting distances along the manifold. So if you think back to that Swiss roll, the distances are as if you unroll the Swiss roll. So now if you cluster in these new dimensions, you can actually cluster data of various shapes in a way that you couldn't just by using k-means, because k-means is something that depends on distance. So you came up with a new, more intrinsic notion of distance, and now your clustering results perform better. So in a lot of ways, this is what understanding the data geometry brings to you. It gives you a new space or a latent representation where you can think of your data, and this new representation can be more informative than the original one you have. And the other one that maybe some people don't think about is that each of these eigenvectors is a harmonic. This also kind of comes from the heat diffusion setting, where these are the eigenfunctions of a discretized Laplace-Beltrami operator. If you don't know what that means, it's the eigenfunctions of a second derivative operator. And so if you think about what has second derivative proportional to itself, those are like sines and cosines. So these form harmonics on a graph, and you can load any signal on this graph to here to perform signal processing. So what that means is if each of these vertices here were cells, then a signal on this graph is your gene, and you could treat the gene as a signal on this graph, and you could kill high frequency components and things like that. Okay, and so one of the canonical things people do in signal processing is taking the Fourier transform so they can do frequency domain analysis. So these eigenvectors, which are the eigenvectors of this normalized adjacency matrix, are actually the same eigenvectors of a similarly normalized, what's called a graph Laplacian. And so these eigenvectors, if you take their transpose and multiply them with any signal on your graph, it gives the Fourier transform, and then you can go back using an inverse Fourier transform because this operator, because it's positive semi-definite matrix, U transpose is the same as the inverse. And so in my lab, we've used these basic framework to come up with a lot of different techniques. I'll go over a few of them, but also mention other related techniques that other people have come up with. One is the minute you have really good distances, you can try to preserve them in lower dimensions, particularly for visualization, which you see is all important in biology. And we actually have two different papers on this. We had FATE, which we came up with in 2019, and multi-scale FATE, which was published in May. So normally you might be like, oh, but both of them are on the cover of this, so that's really cool. But I think if you have a visualization technique and it's not on the cover, then it's kind of embarrassing. So I actually hired very expensive graphic designers to help me with this. So here's the idea. So now we know a little bit of how to learn, intrinsic manifold distances on data, and I maybe convinced you that distances are really important. But the question is, these manifold distances, can we preserve them in low dimensions? Because that would be really useful. And the reason that would be useful here is a simple illustration. If you have this ground truth data that kind of looks like this, it vaguely looks like some kind of branching differentiation process, deliberately abstracted, then if you really want to understand differentiation from this, maybe this cell type deviated in this lineage, and then this continued on, and it split into these two lineages, this kind of underlying generative process, if you want to understand it, it would be good to preserve this structure. But the question is, do most other visualizations actually preserve it? And usually they don't, mainly because they're not thinking about it. They have other things they want to preserve. PCA wants to preserve maximal variance. TSNE and its cousin UMAP want to preserve near neighbors. And so they don't, but we thought that it would actually be a good thing to preserve in low dimensions. Diffusion maps and the diffusion distance, unfortunately, give a very high dimensional representation of the data where different features are new components. But if you visualize two diffusion dimensions, you usually get a very simplified form where you see 2 to 4 trajectories. So this is what motivated us to come up with FATE. FATE uses the process that I already talked to you about, which takes distances, and then it turns them into affinities. But here we saw something very interesting with biomedical data is that there's a lot of deviation in density. So you actually can kill density differences using your kernel. So you can either use an anisotropic kernel or an adaptive kernel. And they both work to do that, because otherwise what happens is every time you diffuse, you'll fall into a density center, like falling into a ditch. It actually creates drift in your nice diffusion process. And so in order to combat that, we have a specific kernel that we call the alpha decay kernel. And after that, you have these diffusion probabilities, and this is where FATE is different from diffusion maps. Now FATE realizes that each data point is contextualized with relation to every other data point using a probability distribution. So then we try to take a probability distribution distance or an information theoretic divergence between these points, and then we embed into low dimensional space using MDS. So just to recap that a little bit, so the step 0 was to get these denoised manifold affinities using your random walks on your graphs. Now each cell is contextualized by the probability to walk to every other cell. So these aren't near neighbors, it's really telling you how it's related to every other cell. And now, since these two are probability distributions, you want to think about how to compare probability distributions. So you could use Wasserstein distance. That originally, before we worked more on Wasserstein distances, would have been computationally inefficient. Instead, we use a divergence. And then we preserve that in low dimensions using a distance preservation method called MDS. So you might wonder, how are these actually related to diffusion distances? And it turns out they're related to diffusion distances because they're related to inner products of diffusion eigenvectors. And then there's this log damping factor. So what the log damping factor does is it increases the importance of things that are far away in informing your position rather than just things that are near. So this is what makes it different than TSNE and UMAP. And also, the other thing is that it's accounting for covariances, or the inner products arising from random walks. So it also will position things differently if things that are far are different versus things that are near. So the two actually play quite nicely with each other. So FATE can visualize this whole thing, whereas diffusion maps can pick out one trajectory on average from each diffusion component. So maybe what this has shown you is that there's many notions of a distance that could be useful for different things. There could be one thing that's useful for clustering, like the diffusion distances. There could be something else that's useful for visualization, like FATE distances. And so there's, go ahead.
Just for clarification, when you show this kernel as a way to define a better distance, there is this epsilon parameter that sort of controls the decay of that.
Mm-hmm.
Maybe you cover that later. How do you choose it?
So the epsilon parameter, we typically use an adaptive kernel. So it's usually the distance to a kth nearest neighbor. And so it's pushed back to k, which is usually something like 10. We usually test the robustness of that parameter, and it's quite robust in that range, as long as your graph is connected. Because if you think about the way this method works, you roughly have to find your near neighbors, and then you can walk out from there to other neighbors. So that's basically what you need your kernel to do, is to find your near neighbors. So how do you use this to get biomedical insight, which is something I talked about? So the first system we applied this to was stem cell development. So you have these human embryonic stem cells. They differentiate into these different lineages. And so PCA actually understands the fact that initially you had one cell type, low variance, and you went into this state that has many, many cell types as you progress in differentiation through the days. Note that the day label is plopped on top of fate. Fate did not know the day label before I put it in. TSNE, it kind of feels free to shatter, usually when there's any sparse region. It does show some kind of cluster structure, but we're actually able to show that Fate, for example, better captures cluster structure also because it's preserving distances. But one of the problems is it actually puts data points right on top of each other if they're very similar. And that's something TSNE doesn't have. It'll kind of blow up the data points so you can see how many data points are in a cluster. So it kind of depends on what you're looking for. And then diffusion maps show a much simplified embedding. Here's a comparison to some others. This is MDS. So it turns out that I said distances are important, but the one you're given in your data is very bad. It gives you basically the worst embedding possible. So it's important to learn a more intrinsic distance, because if you just preserve the distances you got from the data, like Euclidean or something, that's sort of what you get. And downstream of that, our stem cell collaborator, who's now at University of Georgia, was able to analyze these different lineages. And she was actually able to color these with different genes that allowed her to find marker genes and differentiation pathways that lead to a lot of different lineages. And so this is the point I meant about clustering. So the idea is, if in a diffusion operator, it's going to be very hard to jump from here to here because there's very few cells in between, and that's what'll keep the clusters apart. And similar to how PCA is doing it, but it's going to refine the noise within the clusters. But this kind of gave us the idea that we could do a zoomed in version of Fate, which I'll talk about in a second. So we also sought to see what it is Fate kind of preserves. What we'd like it to preserve is a denoised version of manifold affinity. So we created ground truth test cases where we know what the manifold affinity is. It's a clean manifold, so it's actually a shortest path on your data. And then we added noise to the data, rotated it into high dimensions, and it turns out that's very similar to what Fate is better at preserving. UMAP can preserve it maybe sometimes because it initializes with something called a Laplacian eigenmap. So a Laplacian eigenmap also learns the manifold. And if you don't use that as the initialization for UMAP, it turns out to be not better than TSNE. So it's not restricted to this kind of data that I'm showing you, single cell. You can do it on population genomics data. And I think this plot here kind of looks like Bogdan's PCA plot, so maybe we can convince him to use Fate to separate these lineages out a little bit more. These are different geographic regions of these populations, and it separates them a little bit more. Now coming to the teaser that I had, what is this? So this is actually the visualization of what's happening to a neural network when it reaches a minima. So we went all the way to the minima, it converged, the neural network converged, and then we jump out of it a little bit and we generate points along the points in the parameter space that it retraverses. And it turns out, actually, you can just look at this plot and tell when it's generalizing well versus when we're finding a bad minima. This is different ways of sampling this, but what we discovered is when we do this jump and retrain experiment, we can actually tell the difference. So if you're at a good minima and you jump out, you pretty quickly converge back to the same minima, which gives it kind of this flowery shape. If you're at a bad generalizing minima, it'll try to leave. It'll try to escape and it doesn't come back. And when you memorize it, we still don't know why, but it sometimes escapes, but sometimes meanders back after a long time. But this is kind of an interesting way to do that. And the other thing is these kind of shapes that are very connected. You can run topological data analysis. So this is a persistence diagram. You can actually feed this to a classifier to classify different minima at different points in time. So that was fate, but we were kind of happy with it, but we were also kind of unhappy because we were just seeing one level of resolution. What we actually usually wanted to do, or our biologist friends want to do is, oh, can you zoom into this region and I'd like to see more structure there. This was actually a more acute problem with fate because of this tendency to put points right on top of each other. When you zoom in, they do spread apart. So we sought to do a multi-scale version of fate. And in order to do that, we started with data every point by itself, and then we created a filtration process. And this is similar to the kind of filtration they do in topological data analysis. But usually topological data analysis method don't intrinsically operate on a data manifold, whereas this does. So that's the reason is because we're using a diffusion-based filtration on the manifold. And you see what's happening is each of the data points is getting coarse-grained, pulled next to their neighbors, and at each level you're getting a different resolution of the data, different summarization. Okay, so what's actually happening is we're creating a filter. The filter in terms of graph spectral space is something that modulates the frequency components of your data. So if you have a low-pass filter, that means you get rid of the high-frequency components. What it turns out is when you power the diffusion matrix, you're powering every eigenvalue, and these eigenvalues only range from 1 to 0. So it automatically creates a low-pass filter. And the low-pass filter takes off high-frequency variation, so it's killing variation in your data in a very systematic way. You can look at the frequency domain of this filter, and this is what it looks like. It's very dependent on your T, gives you a sharper low pass. And when we do that, we reveal different levels of granularity in our data. And multi-scale fate is actually a clustering algorithm as well as a visualization algorithm. I was having kind of a little back and forth with Kirill the other day where I was like, TC is not a clustering algorithm, and neither is fate. But multi-scale fate actually is because it operates on top of these coarse-grained clusters. So the idea of zooming in here is this is visualizing one level of this condensation. And when you zoom in, you look further and you see additional structure that was not visible here because you're trying to squeeze all this data from all these dimensions into 2 dimensions. So you always lose information and you want to try to regain some of that. So when we were developing multi-scale fate, Akiko Iwasaki, who's a well-known colleague of mine at Yale, said she had tons and tons of data. In fact, she had 80 million cells that were measured on patients who had COVID at Yale New Haven Hospital because she's part of this Yale COVID Impact Project. And some of them we also had longitudinal measurements on that I won't go over. But we applied the condensation process with fate, which we call multi-scale fate, to this data. And what we did was we were able to combine all the different patients. And now we've colored each cell by a smooth version of this. Does this occur in a patient who died from COVID or who survived? And what this is a density estimate of that signal over this graph, which I'll talk about a little bit later. So what you can see is there are certain regions that light up in red. That means that those cells typically came from the patients who died. And so you can see, according to this, there are some specific features that this process picks out that are not canonical populations, but that are subsets of canonical populations that might be predictive of mortality. Not causal, but could be used to predict. And so because we took all these features that occur at all these different scales, we're able to put those into a random forest classifier and classify with about with only 200 something patients, 83% accuracy, whether they were going to live or die from COVID in retrospective data. This actually got us some kind of weird publicity where we went on NBC Connecticut and stuff like that because, we're the death panel that can tell who can die from COVID. But the interesting thing is that we didn't have to do any of the curation of these features. So really one of the things that this kind of analysis does, even at multiple scales, it gives you natural features that you can use, and you can train machine learning systems with it. And this is actually a fate plot of the patients themselves on the basis of distance between these features. So did you have a question? Oh, okay. So somebody kind of had their hand up. So now I'm going to actually switch a little bit from looking at sets of points to point clouds. And I already kind of motivated it because we had patient data from 200 different patients. And each of these patients is really a point cloud. So what we really did in that previous analysis was we combined all that data and divided them into two groups, whether they died or survived. And we did some analysis, but could we look at those patients all separately as individuals and kind of see what happens? And so this is a question that we were very interested in. And we developed a method called diffusion earth movers distance, which is a notion of earth movers distance that's very fast and operates on the manifold graph of the data. And the idea is to use a multi-scale graph density estimate to derive an earth mover's distance. So earth mover's distance, as I already told you, is a way of transporting one distribution to another. Typically, you have what's called a transport plan. This is a joint probability distribution that says how much should you move from this bin to each of these other bins to go from this distribution, which is this marginal, to the other one. But if you want it to be optimal in the sense that you don't want to spend over amounts of effort doing that, so you're penalizing by how far it had to move. So it didn't have to move far, you have low penalty. If it had to move very far, high penalty. And really, if you take the pointwise product of these and minimize this turns into a linear program. You have to run a linear solver to get the solution. Did you have a question? Oh, okay. Usually you can come up with a faster way of doing this. And that's by finding what's called a witness function. A witness function is something that exposes the difference between these two distributions, meaning it's very light, its likelihood, its expectation is very different under one distribution than it is from another, but it has to be one Lipschitz, so it can't suddenly jump up every time there's a discrepancy to be arbitrarily high. And the reason that this works is proven in this Kantorovich-Rubenstein duality, whose proof is actually not that hard. It actually just involves some Lagrange multipliers and algebra from the primal form to show that this witness function that you're going to find has much different expectation. If you maximize that, you get the same distance as you would from the optimal transport. So typically, here's a dual version. Here's kind of how it works. This is one that was defined on images that kind of have 2D structure. Here, the distribution is on the intensity of the pixel is taken as a probability distribution. So what they do is they take the difference between these two, which is like taking the difference between two histograms. But that won't work because it can be very discontinuous. So they decompose that using a limited number of wavelet scales. And because of the limitation of these scales, you suddenly get something that's legal to be the witness function. So it's somehow a smoothed out version of the difference between the two histograms. But you can't use this on high-dimensional data. It was defined on two-dimensional data. And so on high-dimensional data, we worked to come up with a way to do density estimation given this kind of sampling. So here, if you have just two people, you can combine their data and you can create a signal. If a signal of one here means it came from the sick person, and the signal of one here in this blue means it came from the healthy person, for example. And what you can do is you can smooth out this signal to get a kernel density estimate. But we just didn't want to put a Gaussian kernel on it. We actually solved an optimization problem like this, where it had to be smooth based on the graph Laplacian, but still stay faithful to the signal. So this is called a reconstruction penalty, and this is a smoothing penalty. And one of the solutions to this is actually the heat diffusion kernel. So this gives you an analytic solution to coming up with a kernel density estimate. So basically, it's a smooth version of these signals that give you the density estimate. So actually, we've just used that in our MELT paper to find areas of the cellular manifold that were different between two conditions. So, but now in diffusion EMD, we use this idea between people. So we take all these people, we create a joint data graph, we create these indicator signals, and we diffuse the indicator signal using something that converges to the heat kernel, this diffusion operator. So as these diffuse to wider and wider scales, you get more and more smeared out versions. And this is very similar to the wavelet decomposition that was done on 2D. And then you can actually just take a difference between these signals in L1. And this gives you an Earth mover's distance. The really interesting thing about it is that this Earth mover's distance uses manifold or diffusion ground distance, which is pretty unusual. So that means we're estimating the density and the optimal transport right on the manifold. What we were able to show is that it's really fast, but there are a couple of methods faster than it, like cluster tree and quad tree, that just embed the data into trees and take the difference of node populations, but it's much more accurate than those. So in this example, we created fake probability distributions that were centered on different locations in a Swiss roll. Diffusion EMD is able to come up with an embedding. So here each dot is a whole distribution. And the others come up with a more mangled structure there. Going back to the COVID patients, it actually gives a very nice summarization where these are the patients who died and these are the ones who survived, and you can look to see sort of differentiating features between them, potentially using the multi-scale analysis method. So probably the last thing that I'll talk to you about is curvature, since I advertised it in the beginning. How much more time do I have, or am I done with time?
You got 5 more minutes.
5 more minutes, okay, that should be enough. So this is a new work from our lab. It's called diffusion curvature. It's on arXiv. The main idea is to use the laziness of the diffusion to actually compute curvature in your data. So I'm going to have you guys do a thought experiment. And the thought experiment is, imagine you have this kind of UCLA sticker on your computer. And then let's say you're like, oh, I don't want the sticker just on my computer, but I really want it on everything I own. So here's your cereal bowl. Okay, so now take this sticker off the computer and try to put it inside your cereal bowl this way. Okay? Any thoughts on if the sticker would nicely stick there? No. What will happen to the sticker? It will wrinkle. Okay, now how about the other side of your cereal bowl? It's going to stick nicely there. What'll happen? It could rip, right? Okay, and then what about this cylinder? Sticks well, right? So that's actually the idea of measuring curvature in the data. So if you have positive curvature, you have, in terms of a graph, you have more connectivity and it's coming back, converging back. If you have flat curvature, you have this kind of flat surface like you're a cylinder, and if you have negative curvature, you have these paths going from any data point that are diverging. So reconvergent paths, divergent paths, and kind of flat paths. So there has been an attempt to do this directly on graphs. So if you're given a graph like a molecular graph or something like that, you can compute what's called an Olivier-Ricci curvature. It's a measure that's on an edge. It tells you the curvature of an edge. So the way it computes it is, it looks like the distance between the vertices that are the two ends of the edge, and then it takes a little Dirac signal, diffuses it just a little bit, and it looks at the optimal transport distance between those two little clouds. The idea is there would be no difference between optimal transport distance and just the regular distance between these points in a flat space. If you have positive curvature, the center points are further away than the clouds are. If you have negative curvature, it's the other way. Okay? But this notion first involves optimal transport again, which can be kind of difficult. Also, there's kind of normalization problems when you use straight distances and try to compare to optimal transport distances. The second thing is defined on edges. And our point cloud data, even though we've created an affinity graph out of it, the edges aren't really intrinsic. If you use different metrics and different things, you can get different edge structures. And also there's just so many edges because we're trying to connect up this space. So we wanted to come up with a notion of curvature that's point-based and not edge-based. Point or region. We want to say, is this region curved? How is this region curved? So here we sought a classic result from Riemannian geometry called Bishop-Gromov. So the idea here is the same thing as the sticker phenomenon, is that if you have positive curvature, things kind of bunch up. So you have Riemannian, the volume of a ball with the same radius. Your sticker is actually a ball in 2D of a particular radius. The volume is going to be less than if you had constant curvature. So you can do a diffusion version of this, basically, where if you have, if you look at a random walk that starts at a point and you look at the probability that it ends in a random ball of a certain radius after t steps of diffusion, call this quantity Cx, and you look at this quantity as in this is the probability that it's going to end in the ball versus the volume of the entire ball, then this quantity is going to give you something proxy to curvature. And the way we prove that is because in cases of positive curvature, the volume of the ball itself is going to be bigger. So you're dividing by a bigger number, so you'll get a smaller value for the Cx, and vice versa. And so this gives you a notion of curvature that you can apply to point cloud data. So basically, it depends on the volume of the constant sectional ball versus this ball. So in one case, you're dividing by a bigger number. So we have results of diffusion curvature on toy test cases, like this classic, Pringle, which is a saddle, and also on single cell data. In single cell data, one of the things that curvature actually tells you is points where your cellular subtypes diverge, because that's similar to those diverging random walk picture that I had. So those are negative curvature points, and points where there's more steady development are points of positive curvature. But the kind of most interesting thing that happened was that we're again able to look inside a neural network this way. We can actually estimate not just single point curvature, but a whole Hessian by training a neural network to do this based on diffusion probabilities, since we've proved that they contain information of curvature. And then what you can tell of any location you are in your training is what part of your eigenspectrum is still negative. That means have you converged to a good minima or not. And so we're exploring this as we go along. So I'm probably just going to stop there and ask if you have any questions. But hopefully I've shown you that if you think about these basic geometric quantities, you can think of a new space where your data lives. You can do all your old favorite things in that new space, usually. And it can give you insights on data sets. And so let's see. I'm just going to flash this up in case anyone wanted any code of things I've been talking about.